Welcome to the deep dive. Today we're taking on a really important topic, something that touches a lot of lives, the diagnosis of autism spectrum disorder, or ASD.
Yeah, and for a long time now, the way we've diagnosed it has been almost completely based on behavior, on observation, and really the intuition of highly trained clinicians. That's the gold standard, as they call it.
Right, the gold standard. But while it's definitely powerful, it's also, got its issues, subjectivity, potential biases, bottlenecks in the system.
Exactly. And that's our mission for this deep dive, really. We want to compare that traditional clinical path, understand its strengths, definitely, but also recognize where it's maybe falling short.
And compare it to what's coming up now, these more objective, quantitative methods.
Precisely. Things built on deep learning, advanced neuroimaging, even molecular science. It's a whole new frontier opening up.
And this is so critical because, the numbers are significant. ASD affects roughly 1 in every 100 children around the world. So getting that diagnosis right and getting it early is probably the single most important first step for improving long-term outcomes. But we all know about the waiting lists, the diagnostic hurdles.
They're major barriers. So what we're doing here is pulling together insights from a whole range of sources. We've got systematic reviews looking at how stable diagnoses are over time, current clinical practice guidelines.
And also the really cutting-edge stuff, right? Studies using huge data sets like the Abide neuroimaging data, proteomics looking at proteins, even large language models analyzing doctors' notes.
Yeah, all of it. Consider this your shortcut, basically, to understanding where ASD diagnosis is strong, where it's struggling, and honestly, where it's heading next.
Okay, so let's start with that conventional pathway. How is ASD actually defined in that model?
So fundamentally, it's behavioral. It's defined by observed challenges, specifically in social interaction, communication, and also the presence of restricted interests and repetitive behaviors.
Observed challenges. So it really hinges on what the clinician sees.
Critically, yes, because there isn't a standard biological test you can just order, like a blood test for diabetes. In standard practice, the diagnosis ultimately comes down to clinical opinion.
But it's informed by tools, right? Standardized assessment.
Oh, absolutely. Best practice involves a whole team, ideally. You might have a developmental pediatrician, a psychologist, speech pathologist, maybe an OT.
A multidisciplinary team.
Exactly. And they use standardized tools to try and make those observations more consistent. The big ones you usually hear about are the ADO, the Autism Diagnostic Observation Schedule,
Right.
the CARS Childhood Autism Rating Scale, and the ADIR, which is the Autism Diagnostic Interview Revised, usually done with parents.
Okay, so we have these tools. How well do they actually perform? Let's talk about diagnostic test accuracy, DTA, especially in young kids, preschoolers.
Yeah, this is where you immediately see the complexity. When researchers look at how well these tools stack up against the final judgment of that multidisciplinary team, which is, the practical gold standard we have right now, the ADOS generally comes out with very high summary sensitivity. We're talking around 0.94.
Okay, sensitivity, just to remind everyone, that's its ability to correctly identify the kids who do have ASD, right?
Right.
To not miss cases.
Exactly. High sensitivity means the ADOS is pretty good at making sure true positive cases get flagged. It reduces false negatives.
Good, but what's the catch?
The catch is specificity. That's the flip side. The tool's ability to correctly identify kids who don't have ASD, to avoid misdiagnosing.
And how does the ADOS do there?
That's where the data gets really messy, highly variable, and sometimes, honestly, quite concerning. ADOS specificity in reviews ranges wildly. It can be as low as 0.20 or as high as a perfect 1.0.
Wait, 0.20. That's incredibly low. What does that actually mean in practice?
It means that in certain groups of kids, especially complex cases, you're potentially getting a massive number of false positives.
So kids being diagnosed with ASD who don't actually have it.
Potentially, yes.
Yeah.
For example, if you look specifically at studies that included children with intellectual disability, or ID, the ADOS specificity sometimes plummeted, down to 0.20 in one analysis we looked at.
Why would that be?
Because ID often comes with behaviors that can look like or overlap with autism traits. Things like repetitive behaviors, maybe some communication delays. The ADOS might pick up on those behaviors and flag them as ASD, even if the underlying reason is the intellectual disability itself.
Okay, so that makes differential diagnosis, figuring out what is causing the symptoms, incredibly difficult.
Absolutely. It puts clinicians in this really tough bind. If you prioritize high sensitivity, which the ADOS offers, you catch most true cases. But if the specificity is low in your population,
You risk overdiagnosis.
right, which means anxiety for families, maybe inappropriate interventions, and definitely straining already limited resources.
But the alternative isn't great either. If you try to adjust for better specificity.
Then you almost inevitably lower the sensitivity. You risk missing kids who do have ASD, a false negative. And that means potentially missing that critical early window for intervention.
Yeah.
When the brain is most adaptable, maybe between ages 2 and 4.
So it's this constant balancing act for clinicians. Sensitivity versus specificity, a daily ethical tightrope.
That's a good way to put it. And it leads directly into the next big question. How stable is an ASD diagnosis, especially one made really early?
Right. If a child is diagnosed as a preschooler, say before age 6, does that diagnosis actually hold up over time, say a year or more later?
Well, the research here shows, again, quite a bit of variability might surprise people. Reviews looking at diagnostic stability find estimates ranging dramatically.
Oh, dramatic.
Anywhere from about 60% stability, meaning 40% of kids lost the diagnosis, all the way up to 100% stability in some studies.
60%, that seems low. If almost half the kids in some studies don't retain the diagnosis, what does that tell us?
It suggests a few possibilities. Maybe there was a significant rate of misdiagnosis initially in those preschool years, or perhaps some children, maybe through intensive intervention or just natural developmental changes, genuinely grow out of the diagnostic criteria.
Or maybe a combination of both.
Probably a combination. But it also suggests that how the diagnosis is made initially, or perhaps the specific characteristics of the child, really influences whether that label sticks.
Okay, so the diagnosis itself can be unstable, but what about the symptoms? For kids who do keep the diagnosis, do their core autism symptoms actually lessen over time with intervention?
That's another really profound finding. Some major systematic reviews looked at this, tracking symptom severity using tools like the ADOS over time.
Yeah.
And they found virtually no significant change in the total core autism symptom severity scores for many children who retained the diagnosis.
Wait, really? So billions are spent on early intervention, but the core symptom scores, as measured by these standard tools, often aren't changing much.
According to some of these large reviews, yes. The intensity of the core features seemed quite persistent for many.
What does that mean for intervention? If we're not necessarily reducing the core symptoms, what should the goal be?
It forces a really important shift in thinking, I believe. If those core traits, that underlying neurology, seems relatively persistent for many, maybe the primary goal of intervention shouldn't be solely focused on fixing or eliminating those autism traits.
But shifting towards what? Improving function.
Exactly. Shifting the focus towards maximizing the individual's functional success in life. Focusing on measurable critical outcomes like quality of life, adaptive functioning, how well someone manages daily tasks, holds a job, builds relationships, rather than just chasing a lower score on the ADOS.
So moving from reducing autism to improving life outcomes with autism.
Precisely. It's about supporting skills for navigating the world as an autistic person, rather than trying to make them non-autistic. That's a paradigm shift that really needs to filter into clinical practice and how we measure success.
Okay, that's a really crucial point about intervention focus. Now, sticking with the challenges in the traditional system, we absolutely have to talk about systemic barriers, and particularly the issue of gender bias.
Yes, this is huge. It's pretty widely acknowledged now that our fundamental understanding of ASD, and therefore the diagnostic criteria in manuals like the DSM-5, are heavily based on how autism presents in males.
Based on studies predominantly done on boys.
Historically, yes.
Yeah.
And this has created a really strong male-centric view of what autism looks like. You see it in the prevalence numbers. The ratio is about 4 boys to every 1 girl diagnosed in childhood.
Does that ratio change later on?
It narrows slightly in adulthood, maybe closer to 3 to 1, which might reflect some later diagnoses in females. But that childhood disparity is stark, and it's likely due, at least in part, to biases in the criteria themselves. They often prioritize the kinds of externalizing, more obviously disruptive behaviors sometimes seen more in boys.
So the system is essentially built to detect one pattern and might be systematically missing others, particularly in girls and women.
That seems to be the case. Research, including interviews with healthcare professionals, the actual gatekeepers to diagnosis, really highlights the practical problems this causes.
What are the main barriers they report?
One major one is a lack of awareness or understanding of something called camouflaging or masking.
Can you explain what that is?
Sure. Camouflaging is basically the conscious or sometimes unconscious effort to hide or compensate for autistic characteristics in social situations. It might involve forcing eye contact, even when it's uncomfortable, meticulously scripting conversations beforehand, mimicking the social behavior of neurotypical peers.
That sounds exhausting.
It's incredibly mentally taxing. And research suggests it's significantly more common and often more sophisticated in females. The result is that their underlying challenges are much less visible to an outside observer, like a clinician during a brief assessment.
So a girl or woman might present for help, but because she's masking effectively, the clinician doesn't see the typical signs and might dismiss her concerns.
Exactly. Patients, especially adolescent girls and adult women, often report feeling deeply misunderstood or being told you don't seem autistic because they can hold a conversation or make some eye contact, even if it's taking immense effort.
And if the core traits are hidden, that probably leads to other diagnostic errors.
Yes, it feeds right into something called diagnostic overshadowing.
What's that?
That's when a clinician sees a common co-occurring condition, like anxiety or depression, which are very common in autistic individuals, especially females, and attributes all the patient's difficulties to that condition. They stop looking further.
So they say, oh, this is just anxiety, and they miss the underlying autism.
Precisely.
Mhm.
Because anxiety and depression are so prevalent with ASD, it becomes an easy explanation, especially if the autistic features are masked. The clinician makes the common diagnosis and the diagnostic journey stops there, perhaps for years.
And the healthcare system itself doesn't help here, right? The time pressure.
Or absolutely. Short consultation times are a massive systemic barrier. Think about primary care. A doctor might have, 10 or 11 minutes for an initial appointment?
Trying to untangle a complex, potentially camouflaged presentation of autism, possibly mixed with anxiety or other issues, in 10 minutes seems impossible.
It's nearly impossible. It forces clinicians to rely on shortcuts, on their gut feelings, or what researchers sometimes call illness scripts, these quick mental models based on typical presentations.
And those typical presentations are usually the male ones.
Often, yes. So if someone presents differently, more subtly, more internally, like many females do, those illness scripts don't get triggered. The short time constraint makes it incredibly easy for atypical or complex cases to be missed entirely or misdiagnosed.
Leading to potentially decades without the right understanding or support.
Exactly. It shows how the traditional gold standard, despite its strengths, is fundamentally failing a significant number of people whose autism doesn't fit that historical male-centric mold.
It's really clear we're hitting the limits of a purely subjective behavior-based system. Which brings us perfectly to the next part of our deep dive, the promise of more objective quantitative measures. Let's start right down at the biological level with molecular biomarkers.
Yeah, this is incredibly exciting work. It combines proteomics, which is the large-scale study of proteins, with machine learning techniques.
Okay.
Researchers took serum samples, basically blood samples, from young children, aged about 3 to 7. Some had ASD, some were typically developing controls, and they looked for differences in the levels of hundreds, even thousands of proteins.
And did they find significant differences? I heard the scale was quite large.
Stunningly large.
Okay.
They identified 347 specific proteins that were consistently lower in the children with ASD and 394 proteins that were consistently higher.
Wow, over 700 proteins showing different levels. That's substantial. Did they figure out what these proteins do?
Yes, using bioinformatics. They looked at the functions of these differentially expressed proteins, the DEPs, and they found these proteins weren't just randomly different. They clustered together in very specific biological pathways.
Which pathways were highlighted?
Overwhelmingly, pathways related to the immune system and immune diseases. Also, interestingly, pathways involved in cell motility, how cells move, and focal adhesion, which is about how cells stick together and interact with their environment.
Okay, the immune system link confirms a lot of existing research suggesting immune dysregulation plays a role in ASD. What about cell motility and adhesion? What might that imply?
That points towards potential differences in fundamental cellular processes, maybe even during early brain development. How neurons migrate, connect, and form structures could be affected. It suggests a biological difference right down at the cellular machinery level, long before complex behaviors emerge.
So the potential for a diagnostic test based on this must be huge, something objective.
That's the ultimate goal. And the researchers drilled down even further. They identified a very specific small panel of just 8 proteins.
Only 8 out of the hundreds.
Yes, a concise panel. It included things like immunoglobulins, antibodies, and other factors involved in immune regulation like LYZ, cell, even something related to rheumatoid factor.
And how well did this 8-protein signature work as a diagnostic marker when they tested it?
The results were pretty extraordinary. When they fed this signature into a machine learning model, it achieved near-perfect accuracy in distinguishing the children with ASD from the healthy controls.
Near perfect. What were the numbers?
An area under the curve, or AUC, of 1.000, which is basically perfect classification, and an overall accuracy of 95.27% after statistical validation.
That's incredible. A blood test with over 95% accuracy?
Potentially, yes. This research offers a very real pathway toward developing a highly accurate, objective, early-stage molecular screening biomarker, something that could potentially be used even before clear behavioral signs are present.
That would be revolutionary. Okay, so that's the internal biological environment. What about external factors? We also looked at a study on chronic stress using hair cortisol.
Right, using hair cortisol concentration, or HCCC. Hair traps cortisol over time, so analyzing a small hair sample gives you a non-invasive snapshot of someone's average stress level over the past few months, a measure of chronic physiological stress.
And what did they find? Did having an autism diagnosis automatically mean higher chronic stress?
This is where it got really interesting and maybe a bit counterintuitive. In a large group of kids, over 300 autistic and nearly 300 non-autistic children, the diagnosis of autism itself was not found to be a significant predictor of higher chronic stress levels once they accounted for other factors.
Really? That goes against the common assumption that the inherent challenges of autism must lead to more stress.
It does challenge that assumption when they ran the full analysis looking at all potential influences.
Well, what did predict higher chronic stress?
Two factors stood out significantly. Higher hair cortisol was strongly associated with lower family annual income and also with the child being younger.
Lower income and younger age, not the diagnosis itself.
Not the diagnosis, primarily. It suggests that the chronic physiological stress burden we often see in autistic children might not be an inevitable consequence of their neurology alone.
But more related to their environment, socioeconomic factors.
Exactly. It points towards external socioeconomic pressures and maybe other psychosocial factors playing a huge role. A family struggling financially is likely experiencing chronic stress that inevitably impacts the child, whether they're autistic or not. It highlights a really significant health inequity issue.
So interventions need to consider the whole family context, the environmental stressors, not just focus solely on the child's individual characteristics.
Absolutely. You have to address the structural factors contributing to stress. It's a crucial reminder that autism exists within a complex environment.
That focus on complexity is a perfect bridge to our next section, diving into how artificial intelligence is being used to analyze incredibly complex data, starting with neuroimaging.
Right. AI, especially deep learning, thrives on massive, complex data sets. In the autism neuroimaging world, a key resource is the Autism Brain Imaging Data Exchange, or ABIDE. Specifically, ABIDE, the first iteration.
What kind of data is in ABIDE?
Mostly resting state functional MRI, or RS-fMRI. This technique captures the brain's activity while someone is just lying still, not doing a specific task. It lets researchers map out functional connectivity, which brain regions tend to activate together, suggesting they form networks.
Okay, so you get these complex maps of brain connectivity. Why is analyzing this data so challenging for standard AI?
Because the data is inherently four-dimensional, really.
Right.
You have the 3D space of the brain, and then changes over time. You need AI models that can capture both the spatial patterns where the activity is and the temporal dynamics, how that activity changes and relates across regions over time. Standard models often struggle with that duality.
So researchers are building specialized AI models.
Exactly. Hybrid models are proving very effective. One study we looked at used a combination of a 1D convolutional neural network, a CNN, and a bidirectional long short-term memory network, a BIL-STM.
Okay, let's break that down. CNN and BIL-STM, what does each part do?
Think of the CNN part first. CNNs are great at finding patterns in spatial data, like features in an image. In this context, the 1D CNN scans the fMRI signal over time within specific regions to pick out important spatial features or moments.
So it identifies key spatial signatures, the where.
Sort of, yeah.
Yeah.
It helps extract relevant local patterns from the signal. Then the BIL-STM comes in. LSTMs are designed for sequential data for time series. The bidirectional part means it looks both forwards and backwards in time.
So it tracks how brain activity evolves and connects over time, the how.
Precisely. It captures the long-range temporal dependencies, how activity in one region now relates to activity in another region later or earlier. It maps the dynamic flow of information.
And when they combine these, plus something called attention mechanisms.
Right, attention mechanisms help the model focus on the most relevant parts of the data. This combined CNN-BIL-STM attention model achieved really high performance in classifying ASD versus controls based on the fMRI data.
Oh.
Around 94% accuracy and a 0.93 AUC ROC. It really shows the power of designing AI architectures specifically tailored to the complexities of brain data.
We also saw another approach made for ASD. It used an ensemble method.
Yes, Made for ASD took a different tack. It combined connectivity features extracted using multiple different brain atlases or templates, and also included demographic information. It achieved 75.20% accuracy on the full Abide-I dataset.
That's lower than the 94%, but still impressive given the dataset.
Absolutely. You have to remember, Abide-I is data from many different sites, different scanners, different research groups. It's incredibly heterogeneous or noisy. Getting over 75% accuracy on that real-world messy data, distinguishing ASD from controls just based on brain scans and demographics, is a significant achievement. It shows there are genuine, detectable neural differences despite all that variability.
And maybe the biggest payoff from these AI models isn't just the classification accuracy, but the interpretability. They can tell us which brain regions are most important for making the distinction.
Exactly. This is where AI moves beyond just a black box predictor. The models can highlight the specific features, in this case, brain regions or connections, that are most discriminative.
So, what parts of the brain are these AI models consistently flagging?
This is where some really fascinating and perhaps unexpected patterns emerge. One model, which achieved very high accuracy, up to 98.2% on certain subsets,
Wow.
consistently identified regions involved in visual processing as being the most important features for distinguishing ASD from controls. Specifically, areas like the calcarine sulcus and the cuneus, which are primary visual cortex areas.
Visual regions, not the social brain areas we usually hear about, like the amygdala or fusiform gyrus.
Predominantly visual areas in that high accuracy model, yes. This is a really compelling objective finding coming from the data itself.
Was this finding validated?
Yes. Crucially, the researchers cross-referenced these findings with independent genetic studies related to autism and found convergence. This strengthens the case that these visual regions aren't just a statistical fluke, but represent genuine biological markers.
What about other models? Do they point elsewhere?
Other models, including the Made for ASD ensemble, also frequently highlight connections within the default mode network, DMN, as being significant. The DMN is involved in self-referential thought, social cognition, mind wandering, so that aligns more with traditional ideas.
Okay, so we have signals in the DMN, but also this strong, potentially overlooked signal in visual processing areas.
Right, and that strong visual finding lends significant weight to alternative theories of autism, like the enhanced perceptual functioning model.
Which suggests that maybe the fundamental difference in autism isn't primarily social, but starts with how sensory information, like visual input, is processed differently.
Exactly. Perhaps a different way of perceiving the world leads downstream to the challenges in social interaction. The AI findings force us to reconsider the root causes. Are we targeting the fundamental difference or just its later effects?
That's a huge question. Okay, let's shift from the very expensive, complex world of fMRI and deep learning to something much more accessible, eye tracking.
Yes, eye tracking, or ET, is getting a lot of attention because it's relatively inexpensive, it's fast, non-invasive, and you can use it with individuals of almost any age, even infants.
And it gives objective data on visual attention.
Exactly. It measures where someone is looking for how long, how they shift their gaze. It provides objective, quantifiable metrics related to social visual engagement, like how much someone looks at faces versus objects.
How well does it work for diagnosis in very young children, like toddlers, where we want that early identification?
There was a key double-blind study looking at toddlers between 16 and 30 months old. They used eye tracking to measure social visual engagement while the kids watched specially designed videos.
And compared the ET results to expert clinical diagnosis.
Yes, compared it to a confident diagnosis made by experienced clinicians. The eye tracking measure showed really strong predictive power.
What were the accuracy numbers?
It achieved 85.4% specificity, correctly identifying those without ASD, and 78.0% sensitivity, correctly identifying those with ASD. The overall AUC was 0.90, which indicates high discriminatory ability.
That's very promising, especially considering the practical advantages.
Absolutely. Think about the efficiency. The standard ADOSU assessment takes hours and requires highly trained administrators.
Right.
This eye tracking measurement was entirely automated. The data collection is quick, it gets uploaded to the cloud, analyzed automatically, and generates a detailed report in less than 30 minutes.
Less than half an hour versus several hours. That could drastically cut down those diagnostic waiting lists we talked about.
That's the hope. It could potentially be a highly efficient screening or assessment tool to streamline the process.
Have researchers used eye tracking even earlier, before toddlerhood, like in infants?
Yes, there's research looking at infants considered at elevated likelihood for autism, usually because they have an older sibling already diagnosed. One study looked at babies at just 10 months old.
10 months. What can you measure then?
They measured gaze patterns during free play interaction between the infant and parent. Specifically, they counted how often the infants shifted their gaze to their parent's face.
And did they find a difference?
They did. The infants who were at elevated likelihood for autism made fewer gaze shifts towards their parents' faces compared to typically developing infants. A quantifiable difference in early spontaneous social attention.
Okay, fewer gaze shifts at 10 months. Does that predict who later gets an ASD diagnosis?
Here's where it gets nuanced, and it's a really important distinction. While the elevated likelihood group as a whole showed fewer gaze shifts than the typical group, when they later split that elevated likelihood group into those who eventually did get an ASD diagnosis versus those who did not, but were still considered high risk, the frequency of gaze shifts didn't significantly differ between those two subgroups.
Okay, wait. So looking less at faces at 10 months is linked to being in an autism family, but it doesn't necessarily pinpoint which specific infant in that family will get the diagnosis.
Correct. It seems that this particular pattern with used gaze to faces at 10 months might be more of a feature of the familial risk for autism, something researchers call the broader autism phenotype, or BAP.
The BAP. Those subclinical autistic traits often seen in relatives.
Exactly. So it might reflect a general genetic loading or a family trait rather than being a specific early warning sign only for the infant who will go on to be diagnosed. It's a crucial finding for developing truly predictive screening tools. You need markers specific to the individual's outcome, not just family risk.
That's a very important caveat. Okay, this brings us to what might be the most maybe philosophically interesting application of AI in this area.
Yeah.
Using large language models, LLMs, to try and understand clinical intuition itself.
Yeah, this is really pushing the boundaries. It's not just using AI to make a diagnosis, but to deconstruct how expert humans arrive at that diagnosis.
How did they do this?
Researchers took a massive data set, over 4,000 free-form clinical notes written by professionals involved in autism assessments. These are the narrative parts, the observations, the reasoning. Critically, they deliberately excluded all the scores from standardized tests like the ADOS.
So the AI learned only from the doctor's written thoughts and observations, not the structured test results.
Exactly. They fine-tuned an LLM on this text data, essentially teaching it to understand the patterns of language and reasoning that lead clinicians towards or away from an autism diagnosis, based purely on the narrative.
It learned to read between the lines of clinical notes. How well did it do at predicting the final diagnosis, just from the text?
Remarkably well. The LLM framework, trained only on that unstructured narrative, was able to predict the actual final clinical decision, ASD or not ASD, with 89.1% raw accuracy.
89% accuracy just from reading the notes. That's actually a pretty strong validation of the clinical notes themselves, isn't it?
It really is. It shows that buried within that subjective, unstructured language of experienced clinicians, there's a very high-fidelity signal about the diagnostic outcome. The expertise is captured in the narrative.
But here's the really fascinating part. They didn't just use the LLM to predict. They used it to explain, to interrogate which parts of the narrative, which concepts were most important for its prediction.
Exactly. Using the explainability techniques, they could ask the model, okay, based on the DSM-5 criteria for autism, which criteria mentioned in these notes were actually the most influential in driving your prediction towards ASD?
So it could weigh the importance of different diagnostic criteria as reflected in the clinician's own words. What did it find? Which criteria were most distinctive, most autism critical?
The results here really challenge the conventional wisdom. The model flagged the criteria related to restricted and repetitive behaviors as carrying the most weight. Specifically, stereotyped or repetitive motor movements or speech, criterion B1, highly restricted, fixated interest, B3, and hyper or hyporeactivity to sensory input or unusual sensory interest, B4.
The B criteria in the DSM-5, the non-social ones, those were most important.
According to this LLM analysis of clinical narratives, yes. Those seem to be the features that most strongly distinguish the ASD cases in this dataset.
That flips the usual focus completely. We spend so much time talking about the social communication deficits, the A criteria. What did the model say about those?
That was the most striking finding. The analysis suggested that the criteria related to social communication deficits, A1 through A3, actually carried relatively limited autism-critical information within this cohort of referred individuals.
Limited information. How can that be? Aren't social deficits core to autism?
They are absolutely core to the definition, yes. But what this model might be telling us is that once a child is already suspected of having developmental differences and is undergoing assessment, the specific patterns of repetitive behaviors, intense interests, and unique sensory responses are actually more powerful differentiators for confirming ASD compared to other conditions.
Could it be because social difficulties are present in many different developmental conditions, so they're less specific to autism once you're looking at a clinical population?
That's a very likely interpretation. Social challenges are common in ADHD, language disorders, intellectual disability, anxiety. But the quality, the intensity, perhaps the uniqueness of the restricted interests and sensory behaviors might be what really sets ASD apart in the eyes of experienced clinicians, even if implicitly.
So the LLM, by analyzing the clinician's language, surfaced this potentially implicit diagnostic weighting.
Exactly. And it strongly supports those models of autism that emphasize perceptual and sensory differences as being primary, like the enhanced perceptual functioning model we mentioned earlier. It suggests the most distinguishing features might lie in these sensory and restricted interest domains.
If you're a clinician or a researcher, this is huge. The AI is basically saying, your own collective intuition, embedded in your notes, suggests you should be paying even more attention to the sensory and repetitive behavior aspects for differential diagnosis.
It forces a reevaluation. Are the symptoms that often trigger the initial referral, maybe social withdrawal or communication delays, actually the most specific markers for confirming the diagnosis later on? Or are the B criteria the truly defining characteristics we need to focus on more for accurate classification and perhaps even targeted intervention?
A profound insight generated by AI analyzing human expertise. Okay, shifting gears slightly. But staying with complexity, we have to acknowledge that ASD rarely occurs in isolation. Comorbidities are incredibly common.
That's right. The statistics are quite stark. Nearly 70%, almost three-quarters of individuals diagnosed with ASD also experience one or more co-occurring conditions. These can be general medical issues, psychiatric conditions, or other neurodevelopmental conditions.
What are some of the most common comorbidities?
We see high rates of things like epilepsy, various gastrointestinal problems, constipation, reflux, food sensitivities, and significant sleep disorders are very common as well. Plus psychiatric conditions like ADHD, anxiety, depression, and other neurodevelopmental issues like learning disabilities or ID.
So managing these co-occurring conditions has to be a central part of care, right? Not just focusing on the core ASD symptoms.
Absolutely essential for long-term outcomes, which is why clinical practice guidelines strongly recommend a systematic assessment for these things right from the start. It's often called an initial somatic assessment, or ISA.
A systematic check for physical and other related conditions. Who should be involved in that?
Ideally, the guidelines suggest input from pediatric specialists, potentially neuropediatricians if there are neurological concerns, and genetic specialists, a comprehensive team approach.
What kind of basic screenings are considered essential?
Definitely things like hearing tests and visual examinations, as sensory impairments can mimic or worsen ASD symptoms. And systematic genetic investigations are increasingly recommended.
Why genetics specifically?
Because a number of known genetic syndromes can present with features of autism or increase the likelihood of ASD. Identifying an underlying syndrome can have implications for prognosis, management of associated medical issues, and family planning.
Are there routine biological tests like broad metabolic screening recommended for everyone diagnosed?
Generally, no. Routine, broad metabolic testing isn't recommended unless there are specific red flags, things like developmental regression, losing skills, specific physical features, dysmorphisms, or other clinical indicators pointing towards a possible metabolic disorder.
So be vigilant, but don't overtest without cause.
Exactly. The ISA is about being thorough and systematic regarding common and important comorbidities, but avoiding unnecessary low-yield testing. Keep a high index of suspicion for rarer things like metabolic disorders, but investigate based on clinical presentation.
Okay, that covers the clinical complexity of comorbidities. Finally, let's loop back to prediction using large-scale data, this time from electronic health records or EHRs.
Right. EHRs contain a massive amount of real-world clinical data diagnoses, medications, lab results, visit notes accumulating over a patient's life. The challenge is using this data for early prediction of relatively rare conditions like ASD.
Why is predicting rare events early so hard?
Well, if only 1–2% of the population has ASD, even a pretty good predictive model might generate a lot of false alarms if you apply it broadly.
Yeah.
And you're trying to predict it years in advance, say, predicting an ASD diagnosis by age 5 or 6, using only data from the first 18 months of life. That's a tough signal-to-noise problem.
So how did researchers try to improve early ASD prediction using EHR data, knowing that ASD often overlaps with other neurodevelopmental conditions, or NDCs?
They leverage that very overlap. They realize that conditions like ADHD, learning disabilities, global developmental delay often co-occur with ASD and might share early risk factors or precursors in the EHR data.
So they built a model that looks for patterns across multiple conditions simultaneously.
Exactly. They developed a novel machine learning approach called a conditional multi-label model. Instead of just training a model to predict one label, ASD, yes, no, it learns to predict multiple labels, ASD, ADHD, GDD, etc., conditionally.
What does conditionally mean here?
It means the model uses the predicted probability of one condition to help inform the prediction of another. For example, if the model sees early signs pointing strongly towards global developmental delay, it might increase its predicted probability for ASD as well, because those conditions are known to be correlated.
So it uses the known clinical overlaps to make smarter predictions for each individual condition.
Precisely. It breaks down the hard task of predicting multiple rare, correlated conditions into more manageable steps, where evidence for one bolsters evidence for another, reflecting the clinical reality.
And did this multi-label conditional approach actually work better than just trying to predict ASD alone?
Yes, it significantly improved the predictive performance for identifying children who would later receive an ASD diagnosis by age 5.5, using only that early EHR data from the first 18 months.
How much better?
It boosted the AUOC, that measure of overall predictive accuracy, from 0.76 for a standard single-label model up to 0.80 for the conditional multi-label model.
That jump from 0.76 to 0.80 might sound small, but in predictive modeling for rare events, that's quite meaningful, right?
It's a very meaningful improvement, yeah. It demonstrates that by modeling the complexity and the correlations between different neurodevelopmental conditions, we can get significantly better at identifying risk for a specific condition like ASD much earlier using real-world clinical data.
That's another fantastic example of using more sophisticated models to handle the true complexity of the situation. Okay, this has been an incredibly rich journey from the nuances of a 10-minute clinical consult all the way to AI deconstructing brain scans and clinical notes. Let's try and synthesize the key takeaways for our listener.
I think the biggest takeaway is that we're clearly in a major transition period. The traditional subjective clinical gold standard, while still essential, is being powerfully augmented and in some ways challenged by objective, quantitative measures emerging from multiple fields.
Right. We saw that at the molecular level with proteomics identifying immune system protein biomarkers that show near-perfect diagnostic accuracy in early studies. That hints at future screening possibilities even before behavior is the main focus.
Then at the neural level, sophisticated deep learning applied to neuroimaging isn't just classifying ASD. It's giving us objective, interpretable insights into the underlying brain differences, highlighting not just the expected social networks like the DMN, but also, surprisingly, visual processing regions.
Yeah, that visual finding really stuck with me, especially how it aligns with theories about enhanced perception perhaps being more primary.
Then at the behavioral level, we have technologies like automated eye tracking, offering fast, objective, scalable measures of things like social visual engagement, showing high predictive power in toddlers, and holding real promise for streamlining those overloaded diagnostic pathways.
But alongside all this exciting technological progress, we absolutely cannot forget the persistent systemic issues. The traditional pathway is still hampered by very real barriers.
Absolutely. Things like gender bias baked into the criteria, the challenge of recognizing camouflaging, the risk of diagnostic overshadowing where symptoms get misattributed, and the crushing reality of time constraints in clinical practice. These all remain significant hurdles that need addressing through policy, training, and systemic change.
And perhaps the most profound takeaway, for me at least, came from that AI analysis of clinical intuition itself.
The LLM work, yeah. It suggests that advanced AI isn't just going to be a faster diagnostician. It's actually capable of interrogating the very foundations of our diagnostic criteria.
Finding that in the language of clinicians themselves, the non-social features, the sensory reactivity, the intense interest, the repetitive behaviors seem to carry more unique distinguishing weight for ASD than the social communication deficits we've historically centered on.
It's a finding that really compels us to rethink what the most critical defining characteristics of autism might be, especially for differential diagnosis.
Which leads us directly into our final provocative thought for you, the listener, to take away from this deep dive.
If these emerging objective technologies, the molecular markers, the neuroimaging AI, the LLM analyses, continue to converge and robustly indicate that the most diagnostically distinctive features of autism are actually rooted in sensory processing and restrictive patterns of behavior, rather than primarily in social communication deficits.
Then what does that truly imply for the future of intervention? Should we, as a field, be considering a fundamental shift? Perhaps moving the primary focus away from directly teaching neurotypical social skills and instead prioritizing the support of underlying perceptual and sensory needs first.
Could addressing those sensory and processing differences be the foundation upon which more functional communication and a better quality of life can then be built? Should intervention hierarchies be flipped?
It's a radical question, challenging decades of practice, but one that the emerging science seems to be demanding we ask.
It's a fundamental challenge to the current paradigm, and how we answer it will likely shape the next decade of autism research and care.
A truly compelling question sparked by this convergence of clinical wisdom and cutting-edge technology. Thank you for diving deep with us today.